So first of all, I would like to thank John and Tony for the invitation. It's kind of difficult to give a talk after Nick had this firework of ideas. So, but he did introduce a lot of things that I think I can pick up that may make us think about the interaction between the tumor and the immune system. So I come from Bonn, and actually I'm originally a clinician, a dermatologist who has been thinking a lot about melanoma and the immune system from the start of my training. And when I started my research many years ago, I was training in histopathology, and I was thinking a lot about why do these melanocytes transform, and how do these T cells get there? Why do they get there? Something that we still haven't figured out yet. So at that time, we just discovered a first melanoma gene, the same time, 9p21, and then later discovered it to be p16. And what we were fascinated is that we also discovered that these T cells in the melanoma actually do see specifically the melanoma, something that was debated for many years, and actually people in Mainz, when I was then working, had a hard time of convincing others. So what was kind of disappointing is that many of these T cells appeared to see differentiation antigens, because it was at the time thought, as Suzanne pointed out before, that they see all these mutations, these mutated epitopes. However, a friend of mine, Thomas Wölfel, he actually catalyzed my career in the beginning, and he found T cells, and with these T cells identified a mutation in the cyclin-dependent kinase 4. And actually, this mutation, R24C, appeared then later to be in the binding site of p16, and it basically abrogated p16-induced senescence. So actually, it was an oncogenic mutation that was seen by the T cells, and it will be important for my work later. I just want to point out that rarely, but it is found also in the germline of patients. So somehow these T cells did see the melanocytic transformation in a way. So 20 years later, of course, the dream that we had at the time that we could now immunize and get a T cell response going and effective in the tumor tissue has caught on, and I'm really fascinated by the developments of the last couple of years and what we've listened to today. What we've been thinking about a lot is, what about these tumor cells? So when we start as the immunologists in the immunology side, sort of we kind of, like this picture suggests, take this tumor for an anonymous mass. It's some tumor cell that we don't really know much about. If you listen to tumor biologists, they look at the other way. They have a very complicated picture of the tumor, and then the immune cell is an anonymous mass on the outside. So actually, what I would like to advocate is that we have to think more about this interrelationship, and I think that's also what we're catching on now. So as you heard before, adoptive T cell transfer has actually proved initially the concept that T cells can indeed eradicate melanomas. And I don't need to tell you much about this, because we're going to hear a lot about this, that T cells have been adoptively transferred, and you can see here pictures from the group at the NIH, how powerful this approach can be. But what was shown then is that this can indeed work, but it doesn't work in too many patients, and of course with the new protocols it has been improved. So we've been fascinated by many transient responses in these immunotherapy settings. So actually, in a very nice review that Nick wrote more than 10 years ago, there was a discussion of why sort of tumors resist T cell therapy. And there's, on the one hand, the idea that there's a clonal selection, Darwinian clonal selection, of tumor cells that are somehow antigen deficient or can't present the T cell, the epitopes to the T cells anymore because of MHC loss or beta-2 microglobulin loss, and all the things that have been found. What we've now figured a lot more out is that at the same time, the tumor somehow protects itself, so there's a exhaustion or suppression of T cell effector function in the tumor tissue. So the question is, what is really going on? So we actually were thinking a lot about this, and we found that if you wanted to study this, you needed a system where you would integrate research tools of tumor biology and tumor immunology, so you get both sides to look at. So it took us a number of years, and this is still one of the very first models that we have used. It's a mouse that carries, I'll show you this here on the side. It carries the HGF gene, the hepatocyte growth factor, which drives melanocyte proliferation as a transgene. It's a mouse that was made by Glenn Malino many years back, and it turned out that this mouse has more melanocytes and tends to develop melanomas. So actually, we started out with mice expressing this CDK4 mutation, which was identified by Tcells, and Mariano Babasit's group put it into the germline of mice, and also in the germline of mice, this mutation abrogates P16 dependent cell senescence induction, so impairs cell cycle control. Now, if you put these two together, you get a mouse that spontaneously gets melanoma, and as you see later, these mice are exquisitely sensitive to carcinogens, both UV but also DMBA, which comes in handy if you want to generate melanomas that grow with an accelerated and synchronized phenotype. On the other hand, we developed an adoptive T-cell therapy protocol using the wonderful tools that Nick's lab has made over the years. So we've used the PMEL T-cell receptor transgenic mice where the T-cells then see melanocytic antigen, and then we developed a protocol where we transferred these T-cells, activated them with a recombinant adenovirus, and then we conditioned the mice with chemotherapy, and instead of IL-2, we were always, because that's things we were working on a lot, we were activating the immune system with immunostimulatory nucleic acids. So if you do this, an experiment looks like this. So here you see a mouse that develops different melanoma. So when you put DMBA on the HGF-CDK4 mouse, within 60 days, all mice develop several melanomas, depending on the dose. So a single DMBA dose. And the mice is basically synchronized. And another 60 days, more or less, then the mice have tumors that progress beyond 10 millimeters and have to be sacrificed. So this is what, if you chart the largest tumor, there are always a couple of leading tumors. This is about what it looks like. So if you then treat with these T cells at this point, you see here that you can actually control tumor growth for 1 or 2 months, but then tumors eventually relapse and grow. And we couldn't see this in transplantable models before because either it works or it doesn't work. So rarely that in a B16 transplantable model, tumors come up late again. So we thought, okay, these T cells, of course, could have been lost effector functions. They could have totally been deleted. So can we re-stimulate these T cells at the point? And then what we did is we actually re-stimulated them again with virus and immunostimulation. And you can see here that in many cases, you can then prolong tumor control. See this here, tumors go away again, and then tumors come up late. And when these tumors come up late, what we frequently saw is that they now switched phenotype. So the T cells could be effectively re-stimulated. This prolongs tumor-free survival, but then tumors eventually come up. So what we then were investigating, we were doing this in parallel. We did serial transplants and also generated lines from this HGF-CDK4 mouse melanoma. Because in this primary setting, of course, you had several tumors, and you could never be sure, could this one tumor come up again, or was it another tumor that would come up late? So when we were transplanting these tumor lines and generated a line, we also had the possibility to do pre-post. To say it took us about 3 years to generate lines like this because it's even more tedious than in patients. So once we had this line, we could transplant. It takes about 3 to 4 weeks for the tumors to come up, and after 5, 6, 7 weeks, it's a slow-growing tumor. You get these black masses, which morphologically look very similar to the primary tumors. But then you're dealing with a single tumor that you put at this one point. So if you then treat the mice with this protocol that we did in the primary setting, we could control the tumor again. But what we were fascinated about, we frequently saw relapses. And these relapses, again, were morphologically heterogeneous, and you see again here development of amelanotic, unpigmented tumor parts. So basically what you can sometimes see also in patients. So we were actually quite fascinated. So what we then did, we stained for the antigen, and we said, okay, this is probably an antigen loss variant that we have generated. So the beauty is, of course, now you can take the cells out again. You can look at them in the cell culture. And you see here then one of these cell lines that came out of these relapsed tumors, and it's not pigmented. And if you do a Western blot for GB100, GB100 is lost, as expected. We also found, actually a little bit by accident initially, that the other melanocytic antigens are also gone in these lines. So we checked the recognition by the PML T cells, and as you can see here, the tumor cells were not really detected anymore by the T cells, as evidenced by interferon gamma ELISpot assays with sarcomas that are peptide loaded as controls. So then we said, okay, then we have an antigen loss variant. That was actually not so surprising. So we put it in the mouse again, and looked at this tumor, and he said, okay, when we now treat this antigen loss variant, then of course it shouldn't be treatable. So, but when we treated it, it worked again. So we're actually quite surprised that now a T-cell specific therapy would treat a tumor that has lost its antigen. So what we found out, though, if we put this tumor line that had lost its antigen back into a mouse, it would all of a sudden be, again, melanocytic and express GP100 and other melanocytic antigens. So, and that's why basically the T-cell therapy worked, but then when you looked at the relapse lines, they again predominantly lost antigen expression. So basically it was a phenotypic switching back and forth. So what we figured, what we always saw is in these relapsed tumors, there was a lot more inflammatory infiltrate. There were many more myeloid cells, many more of the T cells that were still left. So we wondered, is it now possible that it's not a selection of antigen loss variants, but maybe it's an indirect effect of the inflammatory response? Because if you go back in the literature, there are some papers claiming that inflammation also causes dedifferentiation of melanocytes. So we actually treated our melanoma cells with macrophage or T-cell conditioned medium. And if you do this, within 72 hours, you find that you lose pigment. And what you also find is that GB100 and TRP2 is downregulated. So, and we did, through a couple of experiments that I'm abbreviating here, looked at what could be the most important ingredient of these supernatants. And we found here that it's TNF. And actually, so you downregulate antigen, not only antigen expression, but also recognition by T cells. So basically, in the inflammatory situation, you switch the phenotype of the tumor cells. So what we also did in parallel, we looked at these, being a histopathologist, we looked at these tumors. We said, well, now what is this really, right? So we found that they were much more spindly. And when in human histopathology, you look at spindle tumors that you suspect could be a melanoma, you always stain for this marker NGFR P75. And they turned out to be rather positive for this NGFR marker, these undifferentiated parts. And so this actually is a marker that is on neurocrest-derived cells, and particularly also on Schwann cells. So, but it's also on a subtype of melanoma cells, also sometimes patchy expressed. So we thought, okay, this might be related to the melanoma. And we did a whole genome analyses, and we found that when you basically look at these tumors, you have a complete downregulation of the pigmentation response that would come back in the retransplanted tumors. And if you then compared that with the because in these tumors, of course, you get a transcriptional picture of the whole tumor, including all the immune cells. So if you would focus on the genes that would also be upregulated or downregulated in vitro on the tumor cell alone, you could extract a signature that was upregulated and high level expressed both in the relapsed cell lines and in the tumors. And that included a number of genes that fell in the GO categories, cell adhesion, response to wounding, and so forth. And the overlap is actually, this is an EMT-like signature that's also associated with chemotherapy resistance. It's a mesenchymal stem cell signature, not surprisingly, a TNF and TGF-beta induced gene signature, and it contains a lot of hypoxia-induced genes. So we think this is the response that the tumor tissue shows after being attacked by the T cells. So why this is important is because the marker NGFR, which is expressed on neuroquest and also precursor cells from the melanocytes, it was discussed here as a marker of stem cells. And there was a great debate because the labs of Irv Weissman and the lab of Sean Morrison couldn't agree on whether this was important and a marker of tumor-initiating cells or not. So in Irv Weissman's paper, they already claimed that a lot of the human melanoma cells express CD271, which is NGFR, and they lack then differentiation antigens. And then it could be why T cell therapies fail, because he thought at the time these are then tumor-initiating cells with sort of stem cell-like characteristics. So what we found out, though, is that CD271 is actually a marker that is regulated by inflammatory responses. So I'll show you that in a minute. What we also did is, with my friend Thomas Wölfel, we studied whether this mechanism is relevant in the human system. So Thomas has several systems where he has a human melanoma cell line, and then he has several T cells recognizing different categories of antigen. And the question, of course, is, does an inflammatory exposition change this? So if you take here two, for example, there's a cell line that MZ7 that he's worked a lot with, and then SKML29, which I like very much because SKML29 is actually the cell line from which the CDK4 mutation was identified. And it is a cell line from a patient who in the 70s in New York at the Sloan Kettering Cancer Center was treated with several immunotherapies, had a spontaneous response, and is alive today. So if you treat these melanoma cell lines, so this one is very differentiated, and you treat it with TNF, it loses its differentiation. Same as with SKML29, you have very low levels of melanocytic markers, but you still down-regulate them. And now you can look with the T cells. So if you look at a T cell that sees GB100, then TNF down-regulates recognition. Now, if you then look at a T cell that sees a mutant epitope, you see actually there's no effect on recognition. So it's not antigen processing or presentation. It's just that if these T cells predominate, the tumors can easily escape. If mutated epitopes are seen, tumors can not really escape, at least not in this fashion. So the same thing as you can see here. So with SKML29, there's a melan A-specific T cell clone. That recognition goes down, whereas the T cell that sees the CDK4 mutation in this particular melanoma, recognition even goes up. So you have selective modulation of the differentiation status that impacts upon what T cells can see in the tumor. So, but what we were struck by when we were looking at these experiments was that when we studied cell lines, that actually the cell lines, this is here a mouse cell line, but they're human cell lines that look very similar. They have cells that are here in green, GB100 positive, and in red, NGFR positive, and it seemed to be mutually exclusive. So we're actually quite fascinated that these cells were in a culture dish in a sort of a range of differentiation status. And as I told you before, one can separate these by just cell surface expression. So you can have a GB100 positive or an NGFR positive subpopulation here, and then when you treat with TNF, you actually can convert these. So you can again dedifferentiate, but you can dedifferentiate GB100 positive NGFR negative one to the reverse. And actually this also happens spontaneously. So if you don't do anything to them, they spontaneously establish their own equilibrium. And I was quite surprised that one could see that in vitro, because it also has consequences in vivo, that there might be tumor cells that might also be migrating to metastatic sites, and they lose their melanocytic expression program, and we wouldn't even see it as histopathologists. So you can see here that if you sort out this subpopulation, it actually reverts backwards to a more differentiated pathway. So, and what, of course, we also looked at is the expression of PD-L1, because one of the questions, of course, was TNF, does it change expression of PD-L1? It's an inflammatory marker. And you see here that TNF on these mouse melanoma cells doesn't really, it changes the MHC molecule. Here you see this, the TNF expression here. So there's low level of PD-L1 with interferon gamma, it goes up, and TNF doesn't really change that, whereas both affect MHC class 1 expression. But there are some melanoma cell lines that also upregulate PD-L1 upon TNF. But the more strong marker is interferon gamma. You can see this here in this human melanoma cell line. Or this is with conditioned medium on the mouse melanoma cell lines, showing that the conditioned medium from macrophages, which contains a lot of TNF, that's the major driver of upregulation of PD-L1 and interferon gamma here. So, so they're basically, let's go back to this one. There are basically several mechanisms how the tumor cells react to the inflammatory response that is generated by a cytotoxic T-cell attack. What we also found is when we treated the lines with TNF short-term in vitro, and we looked at the different genes that were changed, you see here the pigmentation genes go down. But what I also found fascinating is that fatty acid metabolic processes go down, and then immune response to migration, wounding, and angiogenesis and blood vessel development came up. And I want to just expound on this a little bit, because what we saw in our relapse tumors was the strong tendency to grow along or around vessels. It's what histopathologists call angiotropic growth. So if you look more closely, you can see here that the melanoma cells basically extend along vessels and cling to vessels. So there's another, we believe, another way how they can phenotypically change their behavior. And this is actually also potentially a reactivation of embryonal phenotypes, because in the development, neural crest cells migrate from their source in the brain or at the neural crest to the periphery on endothelial cells, on blood, with developing blood vessels. So that might also play a role in resisting T-cell attack. So in summary, we were searching basically for the relative importance of clonal selection of antigen-deficient tumor cell variants and suppression of T-cell effector functions. And we think what we wound up with is that there's a sort of a mixture, because inflammation induces what we think is phenotypic plasticity of both tumor and immune cells. So basically, you have pressure that selects antigen-deficient variants on the phenotypic level, not necessarily on the genotypic level. And then tumor cells can adapt to the different situation with their phenotype. And when pressure sort of by the immune system subsides, then you get more heterogeneity. So you have different probably compartments in a tumor, and also might explain why you have areas where you have a lot of T cells, other areas where you don't have T cells. So one can think a lot about this. So there's a recent paper that also brought me thinking a lot about this by stem cell researchers. This is a group of Dick, and this is just by Cornelia Poliak, the review or the news and views. So what they did is they basically investigated colon cancer explants serially in nude mice. And they went to great lengths to look at sort of selection with genetic techniques. It's the first science paper I've read that has 4.5 pages of text in an 85-page supplement with 30 pages of supplemental text. So I don't know how long they had to convince reviewers to believe the story, but what they came up with is that they said that under chemotherapy, for example, you have non-genetic functional heterogeneity. So you have tumors, tumor cell subpopulations, which basically go away and come back without changing genetically, and maybe be in a dormant state and so forth. And it's not necessarily only a differentiation phenotype. It can be also just a state, like, for example, a metabolic state. So this changes our view, I believe, that it's not just selection of genetic variants, but maybe also selection of, let's say, phenotypic plasticity, meaning that cells can adapt rapidly to different states. And we actually, we put this in a review because we think it's of more general relevance. So if you have a tumor before some kind of therapy-induced injury, and that can be a cytotoxic injury, it can be a PD-1 blockade or whatever, so T-cell driven maybe, then you get an inflammatory response. And the inflammatory response will always, the tumor perceives, or the organism perceives this, we believe, as an injury response, like an injured tissue in a way. And so it will try to heal this. So this old idea of tumor as a wound. And then there will be cytokines produced, and these cytokines will then cause tumor cells to change their phenotype as well, maybe also a certain selective pressure. And they will adapt, and that will enable relapse. And we heard yesterday that also for BRAF inhibition, this wonderful work by Roger Lowe and Tony Rivas, that maybe this adaptive process is of critical importance for the sort of relapse of different type of tumors to different types of therapy. And I think that we need to learn a lot about this, because if we want to combine, for example, inhibitors of oncogenic signaling and then T-cell therapies or blockade with immunoinhibitory molecules, I think we have to understand what is happening when we are basically hitting the tumor. So Michael Hölzl, who I wrote this review with, he always says it's sort of, he believes it's like if an asteroid strikes this tumor tissue, and then, you know, the rules are upset and something novel happens. And I think to understand that, mouse models will probably be of critical importance if they model exactly what is happening in the clinical situation. And sort of that's what, in part, we're trying to figure out. So with that, I will leave you and with the group that did the work. So I'm the ambassador for our small group in Bonn, and I thank you for your attention.